This conference will now be recorded.
So let's start the day 2 session of the webinar on Vision Science and Optometry. And we have our first presenter, Dr. Harinder Jaseja from Vellore EEG Center, Gwalior, India. So, Dr. Harinder, you can please click on the screen button so that you will be able to share your screen with us. Dr. Harinder, if you're having any queries, please do let me know so that I'll help you out. So at the bottom, okay.
Allow me to share my screen.
Yeah, now I have done it. I've given you the permission. Yeah.
I got a technical glitch there.
Okay. So yeah, that's perfect.
Yeah, so is my presentation visible to all?
Yeah, it is visible.
Okay, hello and good evening, everybody. This evening, I'll introduce you to a novel theory that we have proposed to understand the pathophysiology of glaucoma and its comorbidity with obstructive sleep apnea. My screen is visible? Is my screen visible?
Your screen is completely visible to us.
Okay.
Yeah.
So glaucoma is a chronic progressive optic neuropathy and the second leading cause of blindness worldwide. It is characterized by loss of retinal ganglion cells, thinning of retinal nerve fiber, and cupping of optic disc. Obstructive sleep apnea is a sleep disorder diagnosed by apnea index more than 15 per hour or by an AHI index 5 per hour associated with snoring, choking, daytime sleepiness, and fatigue. OSA has been shown to be associated with several comorbidities like metabolic syndrome, hypertension, cardiovascular accidents, and glaucoma, which all affect quality of life. First association of OSA and glaucoma was reported in 5 members from the same family over 2 generations. In a meta-analysis with 3 cohort and 3 case control studies involving 2 million to 2.2 million participants, a significant association between OSA and glaucoma was reported. Normal tension glaucoma is more common in OSA. Now, what is normal tension glaucoma? It is where the intraocular pressure consistently remains lower than 21 millimeter of mercury, and the optic disc changes are similar to primary open angle glaucoma with following special features like thinning of neuroretinal rim, flame-shaped hemorrhages of the optic nerve rim, deep focal notching of the rim, and peripapillary atrophy. In another meta-analysis based on 9 case control studies with 1086 and 581 controls, average retinal nerve fiber layer thickness was reduced significantly in patients with OSA. A cohort of 44 patients with OSA and 35 healthy controls were followed up for a year, and a significant and progressive reduction of RNFL was observed, based on which it is proposed that along with the severity of OSA, the duration of OSA is also important. Now, currently there are two theories to understand the pathophysiology of glaucoma. One is mechanical theory. The other is vascular theory that causes chronic ischemia, vascular dysregulation, hypoxemia, oxidative stress, and inflammation. However, both theories are unable to fully describe the whole spectrum. Now, we have postulated a novel theory, the connective tissue theory. The extracellular matrix of lamina cribrosa, composed of elastin and collagen, provides resilience and flexibility and preserves the integrity of the axons. In case of thinning of the lamina cribrosa, the intraocular pressure fluctuations lead to deformation of lamina cribrosa and subsequent ONH optic nerve head changes. The lamina cribrosa, this is our postulation, which partitions the intraocular pressure and intracranial pressure, is a thin tissue, the ECM of which is composed of elastin and collagen, and it withstands the fluctuations in the pressure gradient across it. It surrounds, supports, and preserves the integrity of the retinal ganglion cell axons passing through it. Optic nerve head can be considered as a biochemical structure that is exposed to intraocular pressure related stress and strain, and the loss of elasticity and stiffer connective tissue renders the ONH vulnerable to glaucomatous changes. In cases of thinning of LC, IOP fluctuations lead to deformation of lamina cribrosa and subsequent ONH changes. Microarchitectural changes also observed in LC have shown focal LC defects, increase in beam thickness, reduction in pore size, increase in pore variability, which all lend further strength and support to the role of connective tissue in the pathophysiology of ONH changes and glaucoma. The changes occurring in ONH patients with normal tension glaucoma are not much different from the patients with open angle glaucoma. Characteristically, the LC of patients with NTG, normotensive glaucoma, is thinner than in patients with open angle glaucoma. Electron microscopy has revealed control of elastin molecules and their disconnection, curling of elastin molecules and their disconnection from connective tissue matrix in ONHs of glaucomatous eyes. Now, corneal hysteresis is another measure. It's a measure which shows the cornea's ability to absorb and dissipate energy and reflects the viscoelastic nature of the tissue. The biochemical properties like corneal thickness, rigidity, and hydration all play an important role, and decreased corneal hysteresis has been reported to be a risk factor of glaucoma. In fact, an important factor, as we'll see it later. In patients with ocular hypertension, the risk of subsequent development of primary open angle glaucoma was associated with central corneal thickness. Connective tissue weakness is an important factor that underlies the pathogenesis of some local afflictions of the eye such as pseudo-exfoliation syndrome, myopia, and floppy eyelid syndrome, all of which can predispose the optic nerve head to damage even with normal intraocular pressure. So connective tissue weakness is the important factor rather than raised intraocular pressure. Now, PEX is a connective tissue disorder and is associated with normotension glaucoma, thinner lamina cribrosa, and lower corneal hysteresis. Glaucoma and pseudoexfoliation syndrome is associated with this thing and with anti-G and possesses thin LC and lower CH, corneal hysteresis. Glaucomatous eyes with PEX have thinner LC in comparison to patients with open angle glaucoma despite similar intraocular pressure. Recently, LOXL1 gene has been found to be involved in extracellular matrix formation and its stability. It is a member of the copper-containing lysyl oxidase family and is known to catalyze extracellular cross-linking of fibrillar collagens and elastin. Now, evidence from myopic eyes. Beaver Dam eye study and Mammo eye study have reported higher incidence of glaucoma in patients with myopia. Corneal hysteresis decreases in patients with myopia, indicating the role of connective tissue disorder. Optic disc in myopic patients is prone to ischemic damage at any level of intraocular pressure. Glaucoma and floppy eyelid syndrome is recognized by loose upper eyelid that everts readily. Pathogenesis of FES involves weakness of connective tissue in the eyelid tarsal plate, in which decreased elastin has been detected by immunohistochemical and electron microscopic studies. Now, as you can see in this, the pictures above are of FES patient and normal patient showing the extracellular matrix and immunofluorescence elastin stain. The collagen is also disturbed in FES patient compared to normal. Electron microscopy shows a reduced number of elastic fibers and microfibrils interspersed among collagen bundles in the tarsus. The decrease in elastic fibers in FES, floppy eye syndrome, is probably due to elastolysis by protease enzymes, as increased tissue levels of elastolytic proteases, particularly matrix metalloproteases 7 and 9, have been reported in eyelid tarsal tissues. Corneal hysteresis and corneal resistance factor are significantly lower in patients with floppy eye syndrome, and patients with FES have higher susceptibility to glaucoma and higher prevalence of OSA. So connective tissue weakness in FES may represent a systemic phenomena, not just localized in the eyelid tarsal. And so there is redundancy of tissue not only in the tarsal plate of lateral canthal tendon, but also in oropharyngeal tissues, which may be responsible for OSA. Patients with sleep apnea syndrome are reported to have a lower central corneal thickness that exhibits a negative correlation with hypopnea index. Higher the index, lesser is the thickness. Patients with OSA have lower central corneal thickness, and patients with moderate and severe OSA have lower corneal hysteresis. Elastic fiber depletion observed in this can also explain the thinning of lamina cribrosa and a weakness of trabecular meshwork. In OSA, the suppleness of tissues in upper airways makes them more collapsible, and OSA is common in connective tissue disorders. So a flow chart trying to explain the remodeling of ECM, extracellular matrix. Recurrent episodes of apnea or hypopnea cause intermittent hypoxemia, leading to oxidative stress and inflammation, increased matrix metalloproteases expression, remodeling of ECM, And acute hypoxemia affects ECM remodeling by influencing collagen and MMP's expression in human corneal fibroblast cells. So OSA glaucoma are we have proposed that are linked through connective tissue disorder, and therefore therapeutic approaches supplemented with anti-inflammatory drugs, anti-inflammation because of the intermittent hypoxemia leading to oxidative stress and inflammation, antioxidants, nutritional interventions targeted towards strengthening of connective tissue may at least retard, if not arrest, the progress of glaucomatous changes and its complications. And this is a paper published in European Journal of Ophthalmology. Thank you.
Thank you for your presentation, Dr. Harinder. So yeah, do we have any questions? I guess there are no questions. Thank you. Thanks a lot for your presentation and your time.
Thank you, everybody. Have a good
Thank you.
day.
Yeah, you too. So let's move on to our next presentation. And for the next presentation, we have Dr. Atano Mujamdar from NIMS University, India. So, Dr. Atano, you can click on the screen button to share your screen with us.
Yeah. Have you given me the permission for the screen sharing?
Oh yeah, just give me a second.
Because I cannot.
Okay, that just went offline. So just give me a second. Let me just do it. Yeah, now I have given it.
So I guess it's not working out here. Mhm.
Okay. Okay, maybe is there any technical issue or with any permissions or anything?
Permissions is asking for.
Okay, maybe you can just send an email
Mhm.
to me with the presentation so that I will just present it for you and you can just give a talk on it. I've
Yeah.
sent I've just sent a text
Yeah.
of my email ID so you can just send it to me.
Sure. Jessica, I have got the permission, but I'm not able to share my screen.
Okay, okay, just give me a second. Can you please try it out now?
Yeah.
Yeah, perfect.
Right.
Yeah, perfect.
So is the screen audible? Is the viewable now?
Yeah, your screen is visible.
Right.
That's perfect.
Start with. Yeah. So hi, good evening, everyone. This is Dr. Atanu, and I'm working with the Tata Steel Hospital out over here in India. And we had a wonderful talk from Dr. Harinder over the glaucoma. My topic today is all about the cataract, which has always been a point of interest for all the surgeon, that the incision, the site of incision, the size of incision. And so my talk today includes manual small incision cataract surgery or a standard phacoemulsification surgery. Does the size really matter for the post-operative surgery-induced astigmatism? So coming on to the point of introduction, SICS, the small incision cataract surgery, is a very widely accepted practice technique on the cataract management for both developing and in developed countries. It is simply cost effective and applicable to all ages, grades of cataract, and in some complications, we can do this. We can do it in some of the complicated surgeries too. But on the other hand, if you look for the MICS, MICS are a bit costlier because the instruments are costlier and it has a steeper learning curve and unable to the hard grades of cataract. We are unable to do the MICS. So moving on to the aims and objective, the aim of the study is this was a comparative evaluation of the post-operative efficacy of manual SICS and coaxial phacoemulsification in the patients with the age-related cataracts. The methods, it was a prospective randomized comparative study that was carried out in my department, and we have done it in 150 eyes of the patient who undergone the surgery of cataract were assessed. So the inclusion criteria includes an age group of 50 to 70 years having a grade of 2 to 4 nuclear sclerosis. And the exclusion criteria includes the absence of the preoperative complications like zonular delegates, corneal opacities, uveitis, pterygium, high myopes, etc. And of course, the traumatic cataracts. So coming to the grouping, micro incision cataract surgery, we did it in the 75 eyes. And on the other hand, the standard incision phacoemulsification cataract surgery, we did again for the 75 eyes. The statistical analysis suggested the data suggested that using the repeated methods of analysis was the variance methods as the tested the significance probability. So the result, what we concluded, we did a study on the first day, 7th day, 30th day, and the 90th day of the post-operative assessment we have done. So we didn't find any obvious changes in CCT, ECC, EPT, ECL, and AVE for both the MICS and SICS. But yes, there was a significant difference in surgery-induced astigmatism. With MICS did a very good job. On the other hand, the SICS was a bit unsatisfactory, though the p-value is less than, it's more than 0.5%. So on in conclusion, we revealed that the microincision cataract surgery proved to be advantageous in the short term as well as in the long term. Surgery-induced astigmatism, which is significantly less by the supportive but of the better visual acuity over SICS. Thus, the microincision cataract surgery, being a superior technique, must be promoted in the practice for the better patient's outcome. So coming to the point of discussion, in manual SICS, the incision that is given superiorly is about 8 millimeters in comparison to the MICS, which we just give 2.8 millimeters. due to the large incision size. Post-operative surgery-induced astigmatism is found to be higher. So coming to the next slide is my video which I have performed over here. There will be a comparison between MICS and a phaco. So we are making a comparison. Just hold on for a moment. Allow me some time. Yeah. So this is what we are performing in SICS. Not many of steps are involved in that. This is just your performing in SICS. The copy is being done at this point of time. So just injecting the taken to cover we are hydrating it now. This elastic has been injected at this point of time to save the endothelium. See, in this case, we are giving a large incision so that the lens can easily inserted into this. This is a large incision. Now trying to make a tunnel out of it. So here we can find there is a complication that the pupil is not well dilated. So we are doing asymptotic assessment of time. Case, I remember this was a diabetic phase, so the pupil was not well dilated. With this case, we are doing asymptotic so that we can well dilate the pupil. We again made an incision. This is for the side foot. implant a rexis onto this. We have to have a very controlled handle onto this because if this then we can foot can be ruptured. Here we can find absolutely round rexis has been performed. Just trying to maintain the ACS. Performing some hydroprotection to wash out the pockets. Just making the lens free from the veneers and rotate it in the 360 degrees. Extending our main tube. Now these are the dialing wheels. With the help of the dialing wheels, we can just deliver the nucleus. Some viscoelastics have been injected so that the epithelium can be saved. We are just delivering of the nucleus. So very successfully we have delivered the nucleus, and now some of the extra proteins were just washing it out. I guess yesterday we had a talk on to the IOLs for the cleaning of this postal capsule. So this is what we are doing at this moment. Now we will come to the lens inside. This is lens and we will dial out the lens now. So the lens has been dialed, and we can find that the lens is properly open here. And by this, afterwards, the technique for the SICS is getting completed. So now we are going ahead with the MICS. So you can find out in this MICS how simple is the steps if you have the skill within us. See only one incision, and this is the second. Axis is being performed. The lens is free by this time, and we have rotated the lens to 360 degrees. And now we have started doing the phaser. We are dividing the nucleus. The step is known as divide and conquer. Now we are chopping the lenses. Yes, chopping the nucleus. A very minimum set of power has been used for the surgery. Almost we have completed it. In the lens, which was the lens has been properly. How easy is this technique is all about? The lens has been fixed into center lens, removed the complications, so easy to perform. So we have done it, and thank you everyone for your patience.
Thank you for your presentation, Dr. Ratan, and also for the visual. So yeah, it was really interesting. So do we have any questions?
Yes, just one for me.
Yeah, sure, doctor. Please go on.
Is it actually technically more difficult to operate through a small incision?
Actually, this is all about the skill. If you see, well, when previously we were used to performing the ECCS, and then we step into the SICS, performing Rexis at that point of time, it was, believe me, it was very difficult for us. But as the time moves on, and you keep on trying, and if you keep on moves on, the things gets very easy. So
Yeah.
it's technically, if you do lots and lots of cases, I believe it's a savior of the time. At the same time, definitely there will be less trauma, and yes, the patient will be benefited. This is what I feel personally.
Yeah. Do many of your colleagues already use this technique?
Yeah, we do.
Yeah. Thank you for that.
Most welcome.
So any other questions? I guess there are no questions. So thank you. Thanks a lot for your presentation.
Thank you. Thanks for inviting me here. Thank you. Thanks a lot.
Thank you. So moving on to our next presentation. It's actually from Dr. Victoria, but from Tel Aviv University, Israel, but due to some conflicts in schedule, she's not able to attend. So instead, we'll have Dr. Yisan from Harvard Medical School giving his presentation of yesterday's, because we have missed his presentation yesterday. So we'll just continue with that. So Dr. Eason, please to click on the screen button so you'll be able to share your screen with us.
Hello, hi, thank you.
Yeah.
Thank you everyone for joining the meeting. So I will start share my screen. Oh, how should I do it?
Okay, at the bottom, like beside my camera screen. So please do click on that button, screen, so you'll be able to share.
So yeah, but I click there. I'm not allowed me to do that.
Or maybe that might be some permissions. Maybe you'll have to give some permissions.
Sorry. So I'm not able to do that.
Mm-hmm.
Oh, I need to restart. They asked me restart the meetings. So I have to log out and then restart.
Yeah, sure. We'll be waiting. No problem.
Sorry about that. Is it my
Your
screen?
Yeah, your screen is visible.
Oh, what happened? Okay. So is that okay? It's kind of like a partial screen, right?
It's anyway visible. Yeah, it's visible.
Okay, perfect. So I can start my talk.
Yeah, sure.
Yeah, thank you. Thank you. So my talk will be on the angiogenesis in disease and the development. So I have no conflict of interest to be disclosed. So my work is actually based on the translational research. It's on mainly on the animal models. So first I want to give an introduction for the ocular vasculature and the development. So this is on the left side. There's the two type of the vasculature bed. One is the
ఇంకా మూడు ఉన్నాయి టాక్స్.
ఇప్పుడు ఎవరెవరు ఉన్నారు ఇప్పుడు ఇది అయిపోతుంది ఇది రాలేదు
excuse me, able to hear me?
Yeah, we can hear you.
Okay. Yeah. So sorry. So the two type of vasculature bed mainly in the eye. One is the retinal vasculature, another one is the choroidal vasculature. And then in the middle section is that shows that 3 layers of a retinal vasculature, like including superficial layer, intermediate layer, and the deep layer. And also the bottom part is choroidal vasculature. So on the left is a developmental stage for the mouse, for the animal, for the mouse retinal vasculature development. So for the mouse, actually the vasculature development start after birth. So on the bottom panel, we can show the postnatal day 1, and then the vasculatures and the superficial layer start to develop, and then they continue grow. So they basically after birth. So it's a little bit different from human. So I know this is a vasculature eye diseases. So there are several type of the eye diseases. They have a vasculature dysfunction. So one is called the retinopathy of prematurity. So this is mainly in the preterm infants. So this when the infants give birth preterm, so they will have this disease. So especially when they use the oxygen, they mainly because of the oxygen treatment. So this disease is a biophasis disease with the initial phase of the vessel loss. So they're in and then the second phase is a vessel proliferation, which is a picture shown on the right. So another type of the vascular eye diseases is in the adult, in the elderly, so which is called age-related macular degeneration, and also called it wet AMD. So they basically have choroidal neovascularization in this disease. So the choroidal have abnormal vessel grow into the retina. And also another type of vascular eye disease is called diabetic retinopathy. So it's the most common complication of diabetics, and then it's more frequent cause of vision loss in working age population. So they have like an early diabetic retinopathy and also advanced diabetic retinopathy. So for the early diabetic retinopathy, there's non-proliferative diabetic retinopathy. And the advanced stage, they can start to proliferate. So there are several types of the vascular eye diseases. They all have like a vascular abnormality. So our lab mainly use an animal model to study the mechanism of this several types of vascular diseases. So we basically use mouse model to mimic some aspect of those kind of disease. So the first model we used is called oxygen-induced retinopathy. It's mainly mimic the retinopathy of prematurity, because all the preterm infants, they will need oxygen treatment. So we use oxygen to induce retinopathy. So we basically put the mouse pups from postnatal day 7 and until postnatal day 12. So basically, it's 5 days in the high oxygen to mimic the oxygen treatment. So the concentration we use is 75% oxygen, and then we'll move back to room air, and then for another 5 days. So then the vasculature start proliferate. So the below picture shows the retinal flat mounted. So this is a retina. So we basically cut the corner and make a flat mount on the slides to show and then with the endothelial cell marker to show the vasculature. So on the left part is the normal without oxygen treatment. So basically, excuse me, it's how the normal mouse retina look like. On the right is the oxygen-induced retinopathy. We can see the blue, the dark, the bright green color, so which is labeled with endothelial cell, which is a vascular cell marker. So you can see a lot of endothelial cell proliferated. And also the bottom part show the vessel loss in the retina center. They have like a area doesn't have any vasculature. Basically, it's the phase 1 phase. It's a vessel loss phase. So this mouse model is widely used to study the mechanism for retinopathy of prematurity. So our lab is mainly use this model to study the mechanism, and also for the retinopathy of prematurity. So another mouse model we used to study the vascular eye diseases is called laser-induced choroidal neovascularization. So this model basically uses a laser to burn the Bruch membrane in the eye, and then so there will choroidal vessel that will grow into the retina. So the figure shows. So the bottom part of which basically is we have like a 4 laser spots per eye, and then we can introduce like a 4 laser choroidal neovascularization lesion. So the 4 green part that shows the blood leakage. So we basically give fluorescence inside the eye, and then if I have like a choroidal neovascularization, the fluorescence will leak. So they show the blood leakage from the choroidal neovascularization, which is can say can categorize this phenotype. And then the red spots on the middle bottom figure, so that's stained by the endothelial cell marker, which is when we do see they show some like endothelial cell or positive like a lesion in the retina. And also we have like a cross section to show the big lesion, bigger endothelial cell grow into the retina. So use this several parameter to categorize this mouse model. So we basically use this mouse model to study the age-related macular degeneration mechanism. So because the age-related macular degeneration have choroidal neovascularization. So this is a laser-induced that can only mimic some aspect of the human age-related macular degeneration, because this is not caused by aging, it's caused by the laser-induced. But that's for the neovascularization aspect, still can mimic the human disease. So this is another model we'll try to use. And also in our lab, we also work on some genetic modified mouse model. So a lot of genes, the genetic background, they can mimic some aspect of the human disease. So in our lab, we use one gene called the very low density lipoprotein receptor. In short, is VLDLR gene name. It's knockout mice. So in this knockout mice, they spontaneously grow the abnormal vessel into the retina. actually into the photoreceptor layers. So, and then later on they will grow into the choroid as well. So they basically have like abnormal vessel. It's similar with like acute age-related macular degeneration. So we will use this model to study the mechanism in the AMD. So yeah, so that's kind of like a 3 disease models to study the vascular eye disease that used in my lab. So the questions is how can we stop androgenesis in the eye disease? All these 3 type of disease I just described, so they all have a proliferative androgenesis. So they basically blood vessel grow crazy. So how can we stop those? If we can stop, so that we can treat that disease. But the question is how can we stop? So the first we'll look at in the different cell type in the retinas. So the retina have like so the inside of the retina. So there are 3 layers of vasculature. So and then all those vasculature they coupling with the different immune cells. So like the picture shows here. So they have like so the vasculature here is dark brown color, say the BV. So too small, probably hard to see. So that's the color. So that's endothelius. That's the blood vessels, and also we can tell a bunch of other neuronal cells or glial cells around it. So for example, Muller glial cells, extracells, and then microglia. And then in the superficial layer, they have retinal ganglion cells. And then in the outer nuclear layer, they have photoreceptors. So all those, they kind of like will have a crosstalk with the endothelial cells. So we want to know how those other cells, they will influence the endothelial cell growth. If we target those other cells, if they're able to stop endothelial cell growth or not. So, and then our lab mainly focus on the immunoregulators. So the one of the factors is called a suppressor of cytokine signaling 3. In short, it's a SOCT3. So this is a master regulator, can control the cytokines, and then can express by multiple cell lineage, and then can have like a feedback loop to control JAK STAT3 VEGF pathway. So everybody knows the blood vessel growth. They're mainly contributor. One of the contributor is VEGF. So it's a SOCT3. It's kind of like a pathway. It's a pathway that can control VEGF as well. Because SOCT3 control STAT3, which is a transcription factor for the VEGF. So suppressor of cytokine signaling 3 is a small protein, contain 225 amino acid. It's a K immunoregulator, which play a role in the inflammation and the infection, can regulate the immune response, tissue inflammation, cytokine production, and also macrophage polarization. So in our lab, we mainly use a genetic mouse model. So we knock out these genes in different cell types. So and see if this gene is important for those diseases for the vasculature phenotype or not. So we first knock out the CFOS in the neuronal cells. So use the nesting promoter driven CRE. Basically just knock out this promoter, have use a CRE-LOXP system, which is a method to specifically knock out the gene in certain cell type. So basically we knock out the SOCT3 in the neuronal cells and the glial cells. So and this is the phenotypical analysis. And then we found that if we knock out the SOCT3 in the neurons, and then we can make this androgenesis it's much bigger. So this is a quantitative data. So that indicates SOCT3 have a protective role for those retinopathy if we knock out in the neuron cells. So and then the angiogenesis is grow very large. So that means definitely neuron have an interaction with vessel. If we knock down the factor in neurons, and then they will change the endothelial cell growth. And then also we detected this endothelial cell proliferation. The first we see the blood vessel growth, it definitely changed. So we want to see the detailed information about the in the cell level. So we detect the endothelial cell proliferation, use a proliferation marker. So although we see the vessel grow more, and then we want to see if the individual cell can have like a proliferation. So this is a cross section of the mouse retina, and then we start with lectin is the endothelial cell marker, Ki67 is the proliferation marker. So and then if they co-localize these two marker, which is show like cyan color, so that means proliferative cells is a lectin positive cells, which is endothelial cells. So we do see knockout SOCT3 have a much more proliferative endothelial cells. So that also suggests that the SOCT3 protection role. And then we want to know, so how the SOCT3 control the cell proliferation and endothelial cell growth. So we first tested the VEGF. So because people knows VEGF will control the endothelial cell growth. So we want to see if SOCT3 can control VEGF or not. So first we do the laser capture microdissection. So we basically use the laser capture different retinal layer, and then we say which layers can express the VEGF. So we can say if that is a contribute this phenotype. So in the OR model, we say the retinal ganglion cell layer have much increased VEGF, and also in the nuclear layer have much increased VEGF. So photoreceptor layer have increased trend, but not significant. And then we do see in the knockout mice, in the SOCT3 knockout mice, and then they have a much more VEGF. So that kind of indicated that the SOCT3 control VEGF expression in the neurons. And also we detected there are 3 types of isoform VEGF. We detected all of them, and then we found that two types of the isoform was regulated in the knockout. So and also that's messenger level, and also we did a protein level, which is now with the Western blot showing the protein, the VEGF proteins are regulated in the knockout. So and also we do the immunostaining to show the localization of VEGF. So we do see in the RGC layer and in the nuclear layer, which match with our laser capture microdissection results. So the next question we want to address, so we know SOCT3 can control STAT3, and then STAT3 can control VEGF transcription. So we want to test it if SOCT3 can active STAT3 or not. So we first to check, we do say SOCT3 knockout in have like a phospho-STAT3 is activated form of a STAT3. So we do say if a knockout the SOCT3, and then we say STAT3 get activated. So that means SOCT3 do control activation of a STAT3 in this disease condition. So we actually this literature reported that SOCT3 definitely controls STAT3 activation, but at a different disease condition, we need to confirm if under this disease, our disease condition, if it's the same case. So we do say it is the same case. and also in the protein levels, and also the immunostaining, we show the same thing. So that means SOCT3 in neurons definitely can activate STAT3 and the disease condition, which is a retinopathy condition. And also people also know for when VEGF, another transcription factor for VEGF is a HIF-1 alpha. It's a HIF-induced factor 1 alpha, which is mainly it's a hypoxia related factor. But interestingly, we actually didn't say HIF is different between wild type and knockout. So apparently, SOCT3 apparently in the SOCT3 knockout mice, the VEGF upregulation is not because of a HIF, it's because of a STAT3. So this figure shows how the SOCT3 control the VEGF in the retina and the disease condition through activation of STAT3. So the next question we want to say, which kind of a cell type is be responsible for this event? So since we target neuronal cells, so the neuronal cells in the which is a nesting driven is mainly in the neuronal cell, different type of neural cells. And also previously people reported that the VEGF expression in the retinopathy is mainly a Müller glial cells and exercise. So we want to say if it's the same case in our study, and also if regulated by the SOCT3 or not. So we stain the Müller glial cells, use a CRY-BP, which is a Müller cell marker. So we see the Müller cells, and also we use a reporter mice, which is labeled the neuronal cells. And then we do see the neuronal cells, which is we knock out the SOCT3 and then co-localize with CRY-BP. So that means neuronal SOCT3 deficiency do happen in the Müller cells. So that means in this mouse line, we do knock out the SOCT3 in the Müller cells. So that could be because of the Müller cells could be one of the cell type that control the that for this events. And also the CRY-BP also a marker for exercise. So that basically these two, they're all glial cells. They share the same markers. So that means the SOCT3, we do knock out the SOCT3 in this two cell type. And we want to say if VEGF will be from this two cell type. So we basically stain with VEGF and then stain with Müller glial and exercise marker CRY-BP, and then we merge the picture together, and then we say if they're from same cells. So it's a magenta color, green color, merged become white color. So that means there's the same cells. So that indicates that VEGF secrete the cells that secrete VEGF. It's a CRY-BP positive cells, which is a Müller glial and exercise. So this figure clearly to show VEGF secretion was controlled, was from Müller glial cells and exercise and controlled by SOCT3. Yeah, so that's one of the figure to show, and then this is the cross section, and then for quantitative to show the Müller glial, so which is the strip shape, which is a activated Müller glial, and then so this is the bottom part is the exercise shape, and also so this is a quantitative data to show neuronal deficiency deletion of SOCT3 neurons that active Müller glial cells to produce VEGF. So the GFAP antibody shows activation of Müller glial cells. So if we see the strip, that means glial cells is activated. So that's the activated glial cells produce the VEGF. So this is our working models. So we basically saw the neuronal glial cells can control retinal ganglion, retinal vascular, neurovascular interaction through VEGF signals. So that we can just say go back to these figures, and then we know the Mueller cells and then exercise definitely can control angiogenesis. If we target these two cell types, we can stop angiogenesis. So, and then my lab also interested in the other cell type. So another questions we want to say how the photoreceptor cells, if we target the photoreceptor cells, if we're able to stop the angiogenesis. So we do use the photoreceptor specific knockout and other factors called CFOS. It's also immunoregulator. And then we use the mouse model, it's genetic mouse model, which I described in the beginning. It's a very low density lipoprotein receptor knockout mice. So this mice spontaneously develop angiogenesis and grow into photoreceptors. So in this mouse model, we do see the CFOS, which is a transcription factor, and then they will upregulated in the knockout mice, in the messenger RNA level and the protein levels, and then specifically in the photoreceptor layers. So, and then we want to say if this genes in the knockout mice is upregulated. So, and then we say if we knock down the genes in the photoreceptors, are we able to stop the angiogenesis? So that first we make some virus do the subretinal injection, and then inject this virus, which can knock down CFOS directly in the subretinal space. We try to affect the photoreceptor directly. So this is a picture to show the green color, which is a GFP to show the virus directly affect the photoreceptor layers. And then we do see the knockdowns. The CFOS level was knocked down at the protein level and the messenger RNA level. So we just use this approach to modulate the CFOS and see if we're able to stop the angiogenesis. And this is a figure to show we do see much less angiogenesis in the CFOS knockout, in the CFOS knockdowns retinas. So this is a quantitative data, and also we do see much. If they don't have much angiogenesis, they also have like a less leakage. So that definitely shows if we target the photoreceptor factors, we are able to stop angiogenesis or reduce the angiogenesis. And then we try to figure out what's the pathways CFOS targeted. So CFOS also another transcription factor for VEGF expression. So we tested the VEGF level and then phosphostatin 3 activation, and then we do see CFOS also can control VEGF and then status 3 activation. So this is our work model. So we basically the experiment demonstrated the CFOS in the photoreceptors can control VEGF and then through status 3 pathways. So that would go back to these figures. So we figured out, so all those other types of cells, including Mueller cells, glial cells, and the photoreceptors, and then they all different cell types can control the angiogenesis. So, and then summary is the neuroglial cells can control neurovascular interaction, and the photoreceptor can control neurovascular interaction through CFOS. And also our lab also interested in the microglia. So this project is still ongoing, so it's in the process. Well, yeah, so we're still working on this microglia project. Yeah, so this is my lab member and then also my funding resource and also all the collaborators. So thank you. Thank you, everybody. I'm happy to take some questions.
Thank you for your presentation, Dr. Yi. So yeah, do we have any questions?
Yes, just one from me.
Yes.
Would it be fair to say that the basis of laser treatment for diabetic retinopathy is precisely to kill off glial and retinal and photoreceptor cells in order to stop the demand for oxygen, which generates the new blood vessels to form?
Yeah, I'm sorry, what's your question?
The question is, would it be fair to say that laser treatment of diabetic retinopathy?
Yeah, I think the laser treatment you're talking about it for the laser for the laser treatment for the disease. So that actually is different for the laser we use to generate the disease model. I know people use a laser treatment for some vascular eye diseases, but this is this laser induced choroidal neovascularization. It's different from the treatment. This laser will basically burn the Bruch membrane to make a holes here, and then the choroid vessel will grow into the retina. So this is different from the treatment. I think I should explain that clearly, otherwise people will confused. Are you talking about the treatment for the disease, right?
Yes. So what you're saying is that the method of treatment that you propose is much more subtle than just burning glial cells.
Yeah, I think for the laser treatment, at least I know for the retinopathy people and AMD people use the laser to treat. They basically burn the neovascularization part, and then they can stop the bleeding, right?
Yeah.
Yeah, I think for these lasers, we basically is work on the healthy mice to burn the Bruch membrane. So now at that time they don't have angiogenesis yet. So basically burn the Bruch membrane, and then they will induce angiogenesis grow. They will induce choroidal cells grow. So this is not treatment. Yeah, this is very confused. So yeah, I should explain that clearly. Yeah,
Yes.
thank you for the questions.
Thank you. Thank you.
So do we have any other questions? Okay, I guess there are no questions. So thanks a lot, Dr. Eason. So yeah, we'll just have your next talk shortly. So in the meantime, we have our next presentation by Dr. Geraint Griffiths from the Association of Sports and School Vision Practitioner, United Kingdom. So yeah. Dr. Geraint, you can click on the screen button so that you'll be able to share your screen with us.
Yes. It's my turn next. Got it. Done it. Should be sharing now.
Yeah. Your screen is visible.
Okay. Am I okay to start?
Yes, sure, you can.
Okay. Well, this is a presentation about reading speed. So this is optometry, really, pure vision science, rather than the ophthalmology side of it, which is so important to optometrists anyway. But it's just want to start with the statement, really, that the success of any optical correction can only be properly measured by its effect on the occupation. So you can measure vision till the cows come home and maximize performance on the Snellen chart, but it's no good if that correction doesn't translate into good occupational performance, and that could be anything: driving, computer use, sport, whatever occupation you consider, which has a visual demand. And there are very few occupations which don't have a visual demand. And the problem is in optometry is that we don't measure this. And as a result, because that measurement isn't made, there's a huge deficit in our understanding of the relationship between vision correction and its effect on occupation. I believe, a new field of science which is ready to emerge. Now, arguably, reading is one of the most important and universal occupations, and in a sense, it's a perfect illustration of the need for an effective measure of performance or reading speed. Now, there are measures of reading speed. They're not generally used routinely. In practice, we start with the Snellen chart and then go on to the reading test type. And as long as people can see that, that's pretty well the end of the exercise. We don't actually measure how well they see it or how fast they read it. Colleagues who specialize in this area do have other methods of testing reading speed, and typically this would be a measure of random word recognition. Now, the reason for having random words is that you can test the subject's ability to read these better, because they can't guess the sentence, they can't fill in the gaps because there's no logic to the way these words are written. And that's obviously the reason why reading a passage of text and timing it with a stopwatch is even less effective, because if someone's bright enough, they can actually extrapolate the meaning, they can guess the words in between, and sometimes read them very fast through a level of intelligence. So the problem with both these tests is that often they measure intelligence more than the ability to recognize individual letters. And there's a brilliant example of this. If I can show you what the brain can do with very limited information. If I bring this slide up now. I hope that's come up on your screen. Now that's called typoglycemia, and you can read that fairly fluently. None of the words are spelled correctly, so I couldn't believe that I could actually understand what I was reading, the phenomenal power of the human mind, etc. Now, I'm not a great speller, but the person who wrote that couldn't spell at all, except that they got all the letters in the word that should be there and started it with the correct first letter and finished with the correct last letter. So measuring reading speed is not as straightforward as you think it could be. Now, the clue to what we need to measure was demonstrated in the measurement and the understanding of eye dominance, which I presented yesterday. So you can see that paper on yesterday's program. But from that study in eye dominance, we concluded that there are two primary visual skills in all sports and all occupations. One is the ability to aim, which fixes our position in space, and the other, which is dependent on the stability of the aiming eye, is to judge depth. So those are the two things we need in order to interpret the world in a meaningful sense. The third condition, of course, is where we are on the vertical, but that's easy because gravity sorts that out for us. Nobody confuses up and down, but many people confuse left and right, and which is often related to an unstable aiming dominant eye. So the next question is, what is the primary skill in the occupation of reading? Well, the first clue is that most people read at a fixed distance. So it's not depth perception, although that might vary from one day to the next, depending on the condition of the eyes, generally at one sitting, you read at a fixed distance. It might be fixed distance of the computer screen or reading or other activities. So this is not a depth perception requirement. So therefore, it must be that the primary visual skill in reading is aiming. Now we need to find a test which exercises that ability. The ability, in essence, if you boil reading down to its main requirement, in essence, from the start when you learn to read, it's reading from one character to another along a line of letters which make up a word. It's not individual words, that's not where we start. It's one character at a time, one at a time. Now, the point about that is that any test we develop to measure reading speed must assess this ability to track, if you like, from one individual character to another. Now that, to a large extent, is not intelligence related, so you don't get that confusing variable upsetting the results. Now, once you've measured the ability to track, then if there's a problem with that, a problem in the speed of tracking, then you need to know what caused it. And the other thing which came out of the research in eye dominance is that the visual process, the occupational visual process, consists of very few diagnostic elements of occupational vision ability, very few, and 6 in fact. One is measurement of eye dominance itself, so you know which is the eye or which is the potentially affected eye. The other is vision, measured at high and low contrast with a logmar chart. Muscle balance, how the eyes work together. Fixation disparity, how those two images, when they combine, are fused in the cortical visual cortex. And then refraction itself, whether the person is long or short-sighted. And finally, color preference, which also has a bearing on the degree of the visual difficulty. So what we're aiming for is a test which can identify people with a problem accurately, so that you can refer those people to the normal visual test where these elements of visual performance can be measured and therefore identify the one or more which is the problem, which then of course you correct in the normal way with ophthalmic lenses and prism if it's necessary, if there's a divergence effect. Now this is the basis of the development of the test which I'm talking about today, which is called the comparative rate of reading speed test. So the basic building block is individual characters. That's what we suggested at the start. And these characters ideally should be letters, not numbers. Because whenever we look at a number or a letter, a visual search goes on in the brain in order for us to decide whether that's an A or a B or a C. Now, when you look at a number, there's only 9 numbers to choose from, so the visual search is quite quick. It's much more difficult if it's a letter because there are 26 letters to choose from. So it takes a little bit longer to do that. So these measurements must involve letters. The characters must be letters, not numbers, to make it as difficult and as exacting as possible. Now, the other thing which current reading speeds don't take into effect or don't allow that extra meaning to be brought out of the test is that they use a single font. Now, we know from the Dyslexia Association that very often people with reading difficulties find a Times font, for example, which is a serif font, much more difficult to read. And that's because the letters themselves have little sticky out bits, the serif. So effectively, it tends to merge these letters together, in effect, increasing the spatial frequency at which these letters appear. So spatial frequency is a critical factor. If it's high, it's very difficult to differentiate one letter from another easily. If you have, for instance, a binocular vision problem where the eyes are moving microscopically, the letters tend to merge and become very difficult to read. So a good measure is the Times font. But we need something to compare that to, because we need a control, if you like, and the control in the reading speed test could therefore be something like a Gill Sans font, which is has no serif. These are very cleanly written fonts, and again, reported by the Dyslexia Association to be ideal because they're easier to read. And certainly if you read two paragraphs, one of Times of the same size as one of Gill Sans, you can see visibly the Gill Sans non-serif font is easier to read. Now, the final criterion in the design of the CREST test, comparative rate of reading speed test, was to add color to one of these measures. Now, one of the things which came out of the research into reading speed, into color preference, which again presented yesterday, is that teal is a very interesting color. People who are light sensitive like a teal tint. It makes things easier to read. So what we decided at the outset was to make the potentially easier font to read, which was the Gill Sans font. Let's make it even easier potentially and make it a nice teal color. So it would exaggerate the difference between these two fonts and somehow enhance the effect of the comparison between the reading speed of Times and the reading speed of Gill Sans. Now, what we ended up with is this test. Now, it comes in two forms. On my monitor, that looks that looks like a mirror type. I don't know what the technicalities of that are, but I'd have thought if I show you a piece of writing, it should be visible to you as normal writing. But if it's not, then that's the limitation of the technology. That's the large typeface, which is presented to children and adults sometimes who can't read the small type, which is the N12 type. Now that is more difficult, but you can see the top paragraph is made up of Times font, individual characters arranged to look like words, but the exercise is to read the characters individually. So you've got the Times font at the top, and then the Gill Sans font at the bottom, printed in this teal color. So that was the test which we took on our research in a school in Wolverhampton in the UK. Now this is a school in a socially deprived area of Wolverhampton, so a great school, a school pushing the boundaries in terms of reaching out to kids in this socially deprived area, to the extent that they loved coming to school because home was horrible, basically. At least they could get a decent meal in school. So these are socially deprived children. And what we did was, and just to let you know, this is a secondary school. So we tested children between the ages of 11 and 12 in year 7, and there were 95 subjects in total. So what we did, therefore, was measure their ability to read these characters, one line of characters, which had 60 characters in it, with a stopwatch, and time their ability to read those letters at high contrast, and then repeat the exercise at low contrast on the Gill Sans. Now the results were very interesting, which I hope I can show you with a slide here to make it easier to present them to you. Now, when we compared the reading speed for the whole group, Times versus Gill Sans with the teal color in it, the results were significantly different. So as a group, they definitely read the Gill Sans font faster. Now, in a sense, the Gill Sans font and the Times font, although you're measuring two fonts, you're not measuring a variable, because the idea is, the understanding is that Gill Sans, the teal font, will invariably be read faster than the Times font, because the Times font has these, faster all the same, the Times font has this high spatial frequency, very difficult to look at. So we weren't surprised in a way, or pleased to know, but weren't surprised that everybody read the teal font faster. Now, just out of interest, what we did then was separate the faster times, those subjects who had the teal font, this is the non-serif font, who read it faster than the Times font. We separated those children from the next group of children who very unexpectedly, this was a totally unexpected result, actually read the Gill Sans font, the easy font, slower. Now, initially, it was very difficult to understand the reason for that, because we thought that this teal green color would make them easier to read. But it wasn't. For some of the children, and this was 27 out of the 65 children, 27 out of the 95 children read it slower. So that's 30% of the children. Now, when you extracted the slower times, then the probability that teal is read faster than Times was off the scale almost. You can see 6.84 e to the 16, which means 0.0 and 15 noughts before you come to the 6.85. Highly significant result. But the same applied to the ones who read it slower. They definitely read it slower. There was definitely a relationship between the two fonts, which was significant. And it's still quite significant in normal terms, 0.00045. Now, what was the reason for that? Well, the first thing that occurred to us is that the difference between tints, as in ophthalmic lenses, and pigment hadn't really been taken into account. So when you print a font like Gill Sans in teal, what you actually do is reduce the contrast of those letters. Inadvertently, you make them more difficult to see for those people who have poor contrast sensitivity. So therefore, we assume, or we propose, that those children, those 30% who read the Gill Sans font slower, had a problem with contrast sensitivity. The others who read the Times font faster had a problem with spatial frequency, high density print. In other words, they were the ones who had a binocular instability, but the ones who read the teal slower were more generally children who maybe didn't have a binocular instability, but had poor visual development. And typically these will be children with uncorrected hyperopia or poor accommodation. Many children have poor accommodation, more than I think a lot of colleagues suspect, because it's not measured routinely in practice. So the etiology of these reading difficulties is different. One suggested a binocular vision problem, the other suggested visual deprivation in the developing years. So the true variable in this reading speed test is not the font, it's the contrast. And therefore we were presented almost accidentally with a test which compared the two fonts, because that's important to see who has the greater difficulty, and also gives us a way to differentiate between the etiology of the reading difficulties, either a binocular vision problem or a contrast sensitivity problem. So in one test, a great screening test for reading difficulties, all this information is derived. Now, in this school in Wolverhampton, of course, you can see from the results that there was a high incidence of binocular vision problem, as illustrated by the high frequency of the slow times on the Times font. Now, binocular vision is so interesting, isn't it? It's it gives us the information we need to know about the world, where we are in relation to the world, and particular with reading. If you can't focus on the written word because the eyes are moving all the time, because there's some binocular instability, it will affect everything you do in life, right from reading, or maybe not reading, to understanding and relating to your colleagues. Some of who can't read, you can read, therefore how do you feel about those colleagues? How do they feel about themselves? This is an important informative period which is directly affected by the ability to read. Now, I mentioned earlier on that Wolverhampton was a socially deprived area, and what you understand from that sort of situation is the people who live within that area, whose children these were, coming from broken homes, not fed. Some of these kids were tiny when they should have been much bigger because they hadn't been fed properly. Alcohol and drug abuse within the family, so they were very poorly disadvantaged, if you like. Now, it's quite possible that the parents of these children were similarly disadvantaged in their formative years for the same reasons, because they couldn't read, and they couldn't develop the hand-eye coordination to learn to write because of this binocular problem. So in summary, what I'm hoping to put across is that the rate of individual character recognition, not normal reading speed, has implications for the ability to read and write in otherwise bright children. A big percentage of these children with slow crest times were bright as their contemporaries, their peers. They just needed a pair of glasses to bring them up together. This, if it's not corrected, and this is the optometrist's job, with ordinary specs and or contact lenses for anisometropia, with prism if necessary to correct the muscle balance problem in a dominant eye which is drifting off the subject. If that's not done during this period in their life, it will be a constant drag on their career development, and they could end up low self-esteem in the same situation as their poor parents are, trying to make sense of life when nothing works for them. So that's all I have to say on the subject. I hope it's been interesting. And just to finally let you know that at the bottom there is the citation. That's the paper written on this subject, which goes into it in a lot more detail. You can get that off the internet. If you just copy in the title, it will come up. But I do hope that provokes a bit of interest in the subject, and thank you very much for listening.
Thank you for your patience and your everything, Doctor. It was really informative and interesting. So yeah, do we have any questions? I guess there are no questions. So thank you. Thanks a lot.
Well, my pleasure. My pleasure.
Yeah. So we have our next presentation from Dr. Yi-san from Harvard Medical School. Dr. Yi-san, are you available? Are you online? Let me just check about it. Yeah.
Laura, I'm sorry. So I saw
No,
my
that's
Insta.
no problem.
So
So,
I
yes, Doctor, I've made you the presenter, so you can just click on the screen button so that you'll be able to share your screen with us.
okay. So I do have a good two presentation. So I missed it yesterday. So that one I just actually present the one is for today.
Um,
So I
mhm.
had okay. So I will give another presentation. So the button is disappeared. So the share button is disappeared.
Screen, you can just click on screen. Oh.
Right. So I can what share?
Yeah, like I've made you the presenter and you can share.
Share.
Yeah,
Okay.
it's perfect.
Are you able to see my
Yeah,
screen?
we are able to see it.
Actually, I didn't see my screen right now. Where's the screen?
Just give me a second. I'll just check who's the presenter. Oh yeah, it's actually by you. Your screen is being shared. Yeah, we can see your screen.
Yeah, but I didn't see my screen.
Oh,
So that's very strange.
do you want? Yeah, your
So
presentation is also visible.
that works.
Yeah,
That
that's
works.
perfect.
Okay, so it's my first time to use GoToMeeting. Sorry, I keep continuing to have a problem. So we use Zoom. So I will give another presentation. Basically, it's one of the it's a detailed information I mentioned in my last talk. So basically for this talk, I will mainly focus on the photoreceptor, how the photoreceptor signal control the retinal angiogenesis. Okay, go to next slide. Doesn't let me go to next slide.
Oh,
Cannot go to next slide. I don't know why.
or maybe you can just click on the arrow marks. Is that not working?
Okay. Yeah.
Yeah.
So I have no conflict of interest to disclose. So the first I want to give an introduction about the androgenic privilege. So photoreceptor usually they don't have a blood vessel growth. So only disease condition. So the photoreceptor can have they grow a blood vessel into the photoreceptor. That's why they call it androgenic privilege. So they once have this disease condition, the androgenic privilege is lost. So people right now don't know much about it, why they have androgenic privilege and why it's lost in a certain condition. So my lab is mainly focused on the molecular mechanism to study this phenomena. So we basically use the mouse model. It's a genetic mouse model. So they have like a blood vessel growing into the photoreceptors, which is a very low density lipoprotein receptor knockout mice I mentioned in my previous talk. So in this mouse model, they do have a photoreceptor, they do have a angiogenesis in the photoreceptor layer, and also they have immunocells in the subretinal space, which is not normal. So basically, immunoprivilege also lost at the later stage in the mouse line. So we basically focus, we will study this figure shows the inflammation occurs early without inflammation self-infiltration. So they have inflammation, but they don't have immunocell infiltration. They only have a later stage. So we try to figure out why the photoreceptor doesn't have blood vessel, and then disease condition have blood vessel. So we first do a screening to say which factor be responsible for this phenomenon. So we first do the microarray analysis, use the what type of container with that.
ఇప్పుడైతే ఇస్తున్నారు కదా తనకి
తీసుకో ఇప్పుడు మరి నీకు ఇస్తాను నీకు ఇందులో కొన్ని దానికి నేను చెప్తాను అలాగే నీకు నేను 13 కొన్ని ఇస్తానని చెప్తాను నేను తీసుకో.
Yeah, and our lab found the CFOS either one of the factors which will be responsible to which will be upregulated in this mouse lab. And then we further examine the expression and the localization of the CFOS in this disease model. Basically, we found the CFOS is highly induced in this genetic knockout mice in the photoreceptors. And then we just say if knockout have a high expression, and then if a knockout, if this is the factor that contribute to the angiogenesis, then we knock out this factor. We should say the rescue experiment. We should stop the angiogenesis. So we basically use siRNA target this gene CFOS, and then we do say the subretinal neovascularization number reduced. And also siRNA is not like effective. It's not like a specifically target a photoreceptor, and then we make AAV virus, which is a specifically target a photoreceptor, and then use a certain promoter. So this is a strategy how we use this viral therapy to knock down CFOS, and then to examine the photoangiogenesis. So this is we use a subretinal injection. So this is a picture shows we only specifically targeted the photoreceptor layer, which is showed in green color. And then this CFOS genes definitely was reduced. And then we do see the subretinal blood vessel number is significantly reduced after knockdown the CFOS. So that means CFOS, it is the factor that contribute to the photoreceptor abnormal vessel growth. And also we also observe the blood vessel leakage in this knockout mice. If we knock out, in this knockout mice we have like a very large amount of leakage. And then if we knock out this CFOS gene, they reduce a lot. So this also indicates the photoreceptor specifically inhibited the CFOS can prevent blood vessel leakage. So this is the photoreceptor sensitivity measurement. And then we do see if we get rid of the upregulation CFOS, and then we can improve the photoreceptor sensitivity. So, and this is we further dig out the mechanism. And then as I mentioned in my previous talk, VEGF is one of the target for this factor, for the CFOS in the photoreceptors. So we basically do different isosform expression examination and also time course. So was from the developmental stage and also the location in different cell in different retinal layers. So we do say if VEGF suppression, if we suppress VEGF in this mouse line, and then we definitely can get rid of the angiogenesis. We reduce a lot of angiogenesis in the photoreceptors. So that indicated VEGF-A controlled by CFOS is the driver to photoreceptor abnormal blood vessel growth. So this is a further dig out the mechanism. So basically we found that if in the disease condition CFOS factors go up, and then targeted gene including VEGF-A, L6, and then it's go up. And then that will induce the STAT3 activation, and then the target go up. So this is the pathways we figured out. So in the photoreceptors, how the CFOS control VEGF-A and control photoreceptor abnormal vessel growth. So beside that pathway, we also found the CFOS may also increase the TNF alpha, which is another type of cytokines that will increase the like a cascade of pro-factor response, which is also can control VEGF-A. Basically, this is two factors that CFOS control L6 and then TNF alpha, and then a separate pathways, but they all link to the VEGF. So they're basically two-arm mechanism to showing how the CFOS control VEGF-A in the photoreceptors, and then which is responsible for the angiogenesis. So since this pathway they involve the SOX3, so CFOS go up, SOX3 is going down. To further validate if this pathway is if it's the real case in this disease model, we modulate SOX3 and see if we're able to see phenotype. And then we do overexpress since the SOX3 is downregulated. So if this is true, then we overexpress the SOX3. We make this higher, then we should say abnormal vessel stop. And then we just use virus to deliver the genes, and then we do see much less abnormal vessel growth. So that indicates this pathway is very likely happened in this mouse model. So, and also this is the phenotype. So this figure shows we do overexpress SOX3 in the photoreceptor layers, and then we do see much less angiogenesis. And then this is the protein levels shows if we overexpress SOX3, they do VEGF suppressed. So that's definitely further confirm the pathway. And also in order to say the photoreceptor specificity, we use a special photoreceptor cells. We modulate those genes specifically in those photoreceptor cells and see if that specifically for the photoreceptors. And then we do see the same trend in the mouse. So since CFOS is the responsible gene to control the photoreceptor abnormal blood vessel growth, then if we use small molecules, we should able to suppress the phenotype, suppress the abnormal vessel growth. So this will increase the transmissional value. So we first use some specific inhibitor like small molecule SR, which can block CFOS target binding to the promoter region. CFOS is a transcription factor that will bind to the downstream gene promoter. So if we block the promoter, so the downstream gene, they cannot transcribe the downstream genes. So this is a validation. So we treated the mice with the SR small molecule, and then this is a several downstream genes, they all go down. So that means the CFOS was blocked. And then we do see less abnormal blood vessel in the photoreceptor layers. So this basically is a summary. So in the photoreceptor layer, if we found the C-phosphates go up, and then this is the indicator photoreceptor layers, and then C-phosphates go up, and then the downstream L6 TNF-alpha go up, and then the L6 will control STAT3, and that will control VEGF, and then TNF-alpha will control TNF-alpha IP3, SOC3, and the STAT3 and the VEGF. So it basically in the photoreceptor, C-phosphates can control VEGF and then control angiogenesis. So that basically is a detailed molecular mechanism to show how the photoreceptor control abnormal blood vessel growth in the photoreceptor layer, which is normally doesn't have a photoreceptor, doesn't have a blood vessel. So this is like a summary. And then I will thank all my lab members and the funding source and all the collaborators. And then thank everybody for attention.
Thank you for your presentation, Dr. Eason. So yeah, do we have any questions? I guess there are no questions. So yeah, once again, thank you. Thanks a lot for your time and your presentation.
Thank you.
So let's move on to our next presentation. And for the next presentation, we have Dr. Alba Lucia Marin from Conference Research Group, Visionscience and University del Valle, Colombia. So just want to confirm if Dr. Alba Lucia Marin is available online.
Hi,
Yeah.
yes, I'm here. Good afternoon.
Good afternoon. So yes, doctor, we have made you the presenter. Kindly click on the screen button so that you'll be able to share your screen with us.
It's asking me to grant access to this application.
Yeah, please do give permissions for that.
Okay. I don't have access to the screen button.
Yeah. Now
Okay.
I have given it. Yeah.
Now share.
Yeah, that's perfect. Your screen is visible.
Okay. Okay. Hi, now you can see me.
Yeah.
Good. Okay, so well, good afternoon, and thanks to the organizers for giving me this opportunity. First, I'm going to introduce myself. For some reason, I cannot move forward.
Or could you please try clicking on enter or space button or the arrow marks or yeah.
So as you introduced, I'm from Colombia. I live in the southern part of the west part of Colombia, here in this area. And I am an optometrist from La Salle University, which is located in Bogota. Then I have a Master of Science in Clinical Optometry from the Pennsylvania College of Optometry, which is now called Sanders University. Some years later, I moved to Sweden and I have my PhD degree from the Karolinska Institute. And now I'm back in Colombia and I work at as an adjunct teacher at the Universidad del Valle. I'm part of a research group called Visionscience. I have my own private practice called Visión y Vida. This project, this study that I'm going to present is part of a bigger project which is called Visual Outcomes, Ocular Findings, and Visual Processing Skills after Allogenic Stem Cell Transplantation in Children, which was done in Sweden. The aim of the study is to study visual perceptual skills, visual motor integration, and ocular motor skills in a cohort of children and adolescents who underwent hematopoietic stem cell transplantation in childhood. Also to identify risk factors for poor visual perceptual skills, visual motor integration, and ocular motor performance. The study design was a longitudinal study in which we included 139 children and adolescents who underwent the stem cell transplantation at the Karolinska University Hospital between 1986 and 2012. They went to the Department of Pediatric Ophthalmology in an annual manner to have their ophthalmological and optometric evaluations between 2004 and 2012. The participants were eligible to visual perceptual skills and VMI if they had a minimum of 3 years of age. And for the ocular motor skills evaluation, they were in the need of having a minimum of 5 years of age and at least 1 year after transplantation. These children went through conditioning modes like total body irradiation in a single or a fractionated TBI. The single dose was 10 gray with cyclophosphamide, and the fractionated TBI was 3 gray times 4 with cyclophosphamide or etoposide. Another group received busulfan, and another group received other type of chemotherapy like fludarabine. The methods used for this part of the study were the TVPS, which is the test of visual perceptual skills, and we used 2 types of tests. One for those ones that were under the age of 13, which is the red group, the red book, and those ones who were above the age of 13, which is the yellow and blue book. These, we chose 3. It has 7 battery tests, and we chose 3 of them. And we chose the visual memory, which the child is briefly shown a design, an image, and then has to identify the same design from a set of 5 figures. The test reflects the subject's ability to remember for immediate call. Another test was the form constancy. This task is to identify one design among the others. This design can be smaller, bigger, turned to the right, left, upside down. It just maintains the same shape. And the third test was the visual memory, the sequential memory. And this one, the child had to choose a sequence, the sequence that was exactly the same as the previously shown. Then the next test was the VIRE VMI, the VIRE Visual Motor Integration Test. And that's also to have to evaluate the, among many, the eye-hand coordination. And for the ocular motor skills, we chose the Developmental Eye Movement Test, the DEM, of which we present to a child 3 slides of different rows of numbers, and they have to read them, these rows of numbers, as fast as possible. So we measure the time vertically, and the third one is horizontally. And we make then calculation about ratio among these two sets of times. In the results, here are the population that was included of all the 139 children. We had about 57, 56, and 29 for the DEM, males 45 and 28 for the DEM. The age testing was about between 13 and 15 years of age. The age of the transplantation was around 6-7 years of age, and the time after the transplantation that the children was examined was around 4 and 7 years of age. In this group of children, we found out that there was a very common finding, which was cataracts. So we took that in account for this study. So we found that 55 around children had clear lenses and 29 in the DEM group. Lens opacities were about half of those ones, and cataract surgery had a third of those ones. All of the kids had mainly very good visual acuities. Those ones there was just one child who had was a myope child who had a 0.16, and the reason he denies he didn't like the wearing of the glasses. So we chose to run the battery test since his near vision was good enough for the examination. And the near visual acuity was good in all of them. In this table, I'm going to show you the results that were underneath the 25th percentile of those ones tested. So we have 37 below the 25th percentile in the visual memory, 46 children in the form constancy, 55 in the visual sequential memory, and 46 in the VMI. That corresponds to 36, 45, 60, and 46%. And in the second row, we have a relationship of female and male, and we see that mainly male are below the 25th percentile. In relationship with the conditioning regimen, we have total of 4 of the 11 that were treated with single dose TBI, 6 in the form constancy, 6 in the visual sequential memory, and 7 in the VMI. In the fractionated, there were a little more children, since the number of those ones who were treated with fractionated TBI was a little bit bigger. The busulfan was those ones in 16, 25, 25, and 23. And other chemotherapy, since the group was smaller, also had a smaller number of children underneath the 25th percentile. In regards of the DEM, we calculated the vertical time, the horizontal time, the ratio, and the number of errors. And all these children were 20 children, 31, 31, and 22 were of the 57 that were having the 25th percentile. So we found that there's a rather big number of children who has number of a result of 25th percentile and below. And we also compared with the malignancy and the non-malignancy group of diseases that were included in the group. And we have 46 in visual memory with the malignant, 46 in the form constancy, 38 in the visual sequential memory, 45 in the visual motor integration, and 31 in the DEM. The rest of the group were included in the non-malignant group. Patients with malignant diagnosis perform better on TVPS memory and form constancy and on the VMI. So the disease had now with specifically a significant correlation with the performance of these results. In general, as a summary, the visual acuity was good in the great majority. Patients treated with stem cell transplantation in childhood performed lower than the reference in the subtest of visual sequential memory and VMI. And it was more in female only in visual sequential memory, and male results were low in both. The children and malignancy perform at near normal and better than patients with non-malignant in TVPS memory. And children with non-malignant disorders perform somewhat better in VMI. Regarding the perception and conditioning, there was no significant difference between perception and conditioning. The age at where the stem cell transplantation was performed correlate positively with the results in the TVPS memory. And patients with non-malignant diseases perform better on VMI with increasing age of the stem cell transplantation. Here we have, we found out that there's a good visual perceptual skills are referred to process of organizing and deciphering visual information and a very important school tool for their academic performance. We need an adequate, precise saccadic movements and for reading performance. And it has been shown to relate to learning readiness and academic achievements in reading, mathematics, and writing, especially during early school years. So it's very important to know when are these children transplanted, treated, because it will affect in somehow their performance at school, just to be in case they are all this ability, all these skills might not be ready when they enter school. There's this research, this study that was done in 2008, in which 22 children under the age of 4 years were assessed, and they had this, they went through the TBI. And there was mild to moderate deficits in visual spatial, attention, mental speed, and working memory. There was another study with acute lymphoblastic leukemia who were conditioned with TBI, which showed VMI, a low VMI, attention deficit was present, and a short-term memory. But children in the long run, they showed an average or above average academic results. So based on this, problems with visual perceptual skills, visual motor function, and ocular motor performance in children and adolescents after stem cell transplantation are affected by multiple factors. One of the factors I will say it's the most important one is the young age of treatment. The youngest will be more his school time will be more affected. Disease-specific background and the conditioning regimen has a minor impact. Studies in this area are very rare. It was very hard to find out references to support or deny the hypothesis, with conflicting results on cognitive and long-term outcomes. Further studies on cognitive development and academic achievements are therefore needed. We need some well-defined and disease-specific patient cohorts separating metabolic diseases. Pre-transplantation developmental assessments are very important to know how is the children before and after the treatment and the transplantation. Complete neurophysiological examinations to determine specifically their IQ and other abilities, fixed time of observations, and patient questionnaires on how visual perception affects their daily life. They spend about a year at the hospital without without any schooling, without any contact with their peers. And here's the reference of this study. If you can find an improvement. So if there are any questions, I'll be glad to hear them. Thank you very much.
Thank you, Dr. Alba, for your presentation. It was neat and clear. So yeah, do we have any questions? I guess there are no questions. Thank you.
Thank you very much. Thanks for help attending.
Yeah. So let's move on to our next presentation. So we've just left a presentation in between, and yeah, we have Dr. Victoria Vishnevskaya from Tel Aviv University, Israel. So let me just make you the presenter. Yes, doctor. Please do click on the screen button now. You'll be able to share your screen. Yeah, perfect.
So do you see the presentation?
Yeah, we can see
Okay.
it.
But okay, 1 minute. I cannot move to presenter's view because okay, it's blocked. Now I can do it. Perfect. Okay, so I would like to take 1 minute to just introduce myself. My name is Vishnevskaya Victoria. I'm the head of the ocular oncology service in my hospital, the Shiva Medical Center that is affiliated to the Tel Aviv University. And I'm also the head of the Israeli Ocular Oncology Group, which is part of the European Ocular Oncology Group, and we are also affiliated to the ISOO, the International Society of Ocular Oncology, and I'm really, really happy for the opportunity to present this fascinating syndrome. And I would like to thank the organizer for this wonderful opportunity. So I will talk with you about the oculodermal melanocytosis, or nevus of Ota, which is a syndrome. Oops, I cannot move the. Okay, I can move. Which is a syndrome that was mentioned as all the way back to 1861, but it was named as the nevus of Ota. For some reasons, okay, I find it hard to forward my slides. It was named the nevus of Ota, or oculodermal melanocytosis, in 1939 by a group of researchers from Tokyo. It's a congenital condition where there is hamartoma of the dermal melanocytes, which means that mature dermal melanocytes are sitting in the wrong location. And it causes a discoloration of the skin, a blue-gray discoloration of the skin of the face in the involved area, and usually it's branches of the fifth nerve, the trigeminal nerve, either branches 1 and 2, or sometimes even branch 3. And it is usually unilateral. It is rarely bilateral, but I will show you some cases that are bilateral. And it could involve not only the skin, which is obvious and everyone can see when they look on their peers, but it could also involve the ocular surface entirely, the mucosal surface of the nose, of the mouth, and even of the brain, leptomeningeal melanocytosis. And the pathophysiology behind this condition is a failure of the melanocytes to fully migrate from the neural crest where they were born as when we were embryos to the epidermis during the embryonal stages. And therefore, we have melanocytes that are actually living located deep in the skin. And this is how it looks. You see the melanocytes deep in the skin where they should be. They should be here on the epithelium of the skin, on the basement membrane of the skin, but in fact you find them in the dermis and even deeper than that. and that is, of course, pathological. So usually when we talk about nevi on the skin, regular nevi on the skin, they usually carry or associated, they carry mutations or are associated to mutations in the BRAF and NRAS genes, the MAP kinase pathway. But these melanocytes are weird because they carry association to the G complex protein, GNAQ and GNA11. And these proteins actually are associated with uveal melanoma. So it's a sort of hybrid between congenital skin problems and congenital uveal melanocytes problems. So the skin condition that are associated are as follows. This is the nevus sofota. You can see the blue deep discoloration of the face. And these are deep, smaller discoloration like blue nevi, and some very extensive discoloration like the Mongolian spot on the buttocks here, or the Mongolian spots on the back of this patient. They all are associated with the same migration of melanocytes. So what is the epidemiology of this condition? So this is a very rare condition. In fact, it is most frequent in the world in the Asian population. The prevalence in the world is about 0.2 to 0.6%. But in Japan, we know that the prevalence could be as high as 1% of the population. But it's still considered a very, very rare condition. And we know that there is a predilection to female. And if we're talking about Japan, which is about 1%, if we look at the male-female ratio, we can see that female are almost 5 times higher in incidence. But on the other hand, we all know that female are more sensitive to aesthetical conditions, and there may be a bias. Maybe there is actually higher prevalence among men, but they do not come to the medical they don't end up in going to medical attention. So usually we talk about a congenital situation or condition, and that's for about 50% of the cases are actually diagnosed right after birth, and all the others usually are diagnosed until the age of 20. And we'll see in a minute why there is a lag in diagnosis. And we also think that there are trigger factors that actually emphasize the condition and make the condition more pigmentary in presentation. And the triggering factors could be sex hormones, exposure to UV light, and maybe some infection conditions. So let's look at the clinical presentations. So first of all, you would see increased pigmentation with age, usually during the puberty time, and usually the post-menopausal stage as well. And you can see that it is very hard to ignore the massive pigmentation. And as we said, the melanocyte could involve not only the skin, but also some internal mucosas. Now I want to emphasize not on the skin, but on the ophthalmic or the ocular presentation, because this will be the focus from now on. The ocular presentation could be limited involvement, which I'll show in a minute. It could be sectorial, only anterior segment, only a part of the eye, or full involvement, including the anterior segment and posterior segments of the eye. And here you can see the different colors of the choroid reflected behind the retina. This is a normal eye, and the left eye is the eye that is involved with the condition. So let's go further and look at the small details. So when we look on the surface of the eye, we can actually see two lines of colors. There is a deep dark color that is going to be the melanocytosis of the sclera, and there is a more brownish color that is more superficial, and you can actually move it with a Q-tip. This is called melanosis of the conjunctiva, and we know that melanosis of the conjunctiva is has higher association to conjunctival melanoma, whereas melanocytosis is usually a benign condition. Now let's look at the iris. This was already published in 1995. These are called iris nodules or iris mammillations, and they are actually made of hamartomatous cells on the iris surface. And if we'll go deeper and look at this at the angle, the angle could also be involved. So if we would like to classify the condition, we will have to look at the classical classification of the skin condition. The classical classification was published in 1939. Tino published it, and it is actually depending on the involvement of the skin. And we wanted to actually add to this classification because as ophthalmologists, we want to emphasize the eye. And this classification actually does not look on the eye. It looks only on the skin involvement. So why do we want to look at the eye specifically? Because this condition has increased risk with the association with ipsilateral glaucoma in the same side. About 10% of those patients will develop glaucoma, and malignant melanoma, either of the conjunctiva, iris, or the whole uvea, choroid, etc. And these risks are very important to us as ophthalmologists. So the largest case series of patients with nevus of Ota with melanoma, uveal melanoma, was published in 1998, and that series included 44 patients with choroidal melanoma, 12 with ciliary choroid melanoma, and 2 with iris melanoma at the median age of 60. And the prevalence, the lifetime prevalence, was 0.26%. And therefore, the recommendation of that study was that these patients need to have a lifelong follow-up that includes twice a year examination, full examination, including ultrasound, to allow an early diagnosis of transformation to melanoma. So at the beginning, we looked at our cohort of the first cohort of 26 patients, all Caucasians, and we wanted to see which of the patients would develop melanoma and glaucoma. And we saw that during the follow-up, there was only one patient who transformed from benign condition to melanoma, and 2 patients developed glaucoma. Based on that cohort, we actually decided to follow the patients and see if we can maybe establish some kind of grading that could help us to decide which patients are in higher risk for melanoma and glaucoma development. And I just want to show you the spectrum of the presentations we saw in the first cohort. This is a lady, this is case number 15. She's a 55-year-old female, and you can actually see that she has dark discoloration around both of her eyes, including the eyelashes, the sclera, the iris that is very, very dark on both eyes, including the choroid, the nose, you can see the nose here, and even the zygoma in the temporal regions. And here you can see the surface of the eye that is heavily pigmented. So this is in the Tino classification. This is stage 4 because it's bilateral. But actually you can see it even goes to posterior auricular. It's a very, very extensive pigmentation on actually a Caucasian patient. And on the other hand, the other extreme is actually a sectorial, very minor melanocytosis in a patient 23. You can actually see a moderate or even subtle pigmentation of the iris, and you can actually go by through it because the surface is not involved. But if you look deeper, you could see that this melanocytosis actually continues with the angle. Here you see a massive angle involvement, and even the choroid, the inferior choroid, is involved. So this would be involving several tissues of the eye, but only in sectorial way. So another finding, very, very interesting finding that was never mentioned before, that we found in our first cohort, is the fact that some of these patients actually lose the crypts. These are the crypts. You see the deep crypts of the normal iris, and these are the nevus of Ota irises, and you see that they lose their crypts either entirely, including the collarette. This is the collarette. Here we don't actually see the crypts, or partially, this is the normal eye of this patient, and on his OTA eye, you can see that he still has crypts, but only on the color. So we were actually very, very intrigued about this interesting condition, the fact that it could involve the whole eye, just a segment of the eye, and sometimes not all the tissues of the eye. And this is another example of partial loss of the crypts. And another one. Being an ophthalmologist and a specialist in ocular oncology, my main interest was actually finding the risk factors for malignant transformation. But also, we wanted to see which of the patients will develop glaucoma. And we looked at the literature again, and we actually saw that there is not a lot mentioned about it, but there is one article by the Shields that saw association between the melanoma-related metastasis of patients with melanocytosis, and they actually show that melanocytosis of the iris, choroid, and sclera carry risk. The highest risk is of the iris, but the melanocytosis of the eyelid actually does not carry a risk for metastatic disease. And that is logical because, as we said, the eyelid are positive for NRAS, and only the other parts are positive for GNAQ and GNA11. And then we also searched for the literature for another tips, and we found another article that examined the thickness of the ciliary body of those patients to see if this is a risk factors. So at that point, we decided to collect our second cohort of patients, and we looked retrospectively on their demographic data, but we also did something prospectively. We took those patients and compared them to maskly, without knowing who is who, to age and gender matched control. And this is our result. We could allocate 40 patients. As you can see here, our ratio is not 5 times higher, but still the female ratio is higher, and the median age was 35. And this is the TINO classifications, which we are not actually interested in. But we look at this cohort and we saw that only 3 patients, now it's 40 patients, not the first 20. 3 patients developed melanoma, malignant melanoma. They were all treated with brachytherapy with iodine, and the tumor was controlled. And all the patients with glaucoma were controlled locally. And then we did a mask imaging comparison between our patients and, as we said, control patients. First, we look at the whole group, and we saw that in the OTA patients, the choroid is thicker, but it was not statistically significant. And also the iris is thicker, but it is not statistically significant. And then we even divided it to patients with iris murmurations and without, and patients with iris lost. And we saw that there is a trend toward thicker irises, but it was not statistically significant. And we again looked at the patients with loss of the crypts, either full loss or partial loss, and we could see that about 60% of the patients have some degree of iris loss. Most of them has a full iris loss. So this could be another risk factor, and we didn't look at it. Now, another important finding or important suggestion we made in this study was a new classification to the ocular condition that was not actually present yet. And we wanted to base our classification on the ocular involvement. I mean, the parts that are involved, either anterior surface, iris, and cord, I'll show it in a minute, and the extent of the involvement. Is it one quadrant or the whole globe? So we divided it to the involved part. Number 1 would be surface only, which means conjunctiva. Number 2 will be surface and iris, 3 surface and choroid, etc. And we actually published this classification in British Medical Journal of Ophthalmology. And this is how the board of classification looks, and I'm going to show it to you in detail. So number 1 will be just the surface, and this is an example of a patient where only the surface is involved with the melanocytosis. And then we look at the quadrant. If it's one quadrant, it's going to be A. If it's two, B. And then if the whole globe, it's going to be D. And then stage 2 will be a patient who has involvement of the iris. And if the surface is involved, we'll call it iris plus, because the iris is not alone, but also the surface is involved. And again, one quadrant is A, and all 4 quadrants is D. And then if we have choroidal involvement, we would call it number 3, and plus for choroid surface. And if there is involvement of the choroid, the iris, and together we'll call it 4. Quadrants are quadrants, and if the surface is involved, we'll call it plus. And here you have an example of 4, because it's both the choroid and the iris. C, because one quadrant is not involved, and it's without the plus because the surface is not involved as well. So we had all the combinations that we could find included in this classification. And here again, you can see the sectorial patient I demonstrated before, and you can see this is a 4A because he has iris, he had the choroid, I showed to you before, but the surface is not involved, so there is no plus, okay? This is the iris, continues with the angle, and going all the way, extending all the way to the choroid, okay? And then we looked at the ultrasound thickening, and we couldn't find any differences. So if I would like to conclude, nevus of Ota is a rare congenital condition. This is actually an hamartomatous condition of dermal melanocytes. It has variable presentations. It has malignant transformation, and those patients need a long life follow-up. We offered in this article and in this research a new ocular classifications, and we are calling for a wider international study to validate this classification and see whether the trend we saw in ultrasound toward thicker iris and thicker choroid is actually a real one, or if we can show that it is statistically solid, we would need for that much more patients than the 40 we had. So thank you for your attention, and I would like to thank again the organizer for the opportunity to share this study with you. Thank you.
Thank you, Dr. Victoria, for your presentation. It was informative. So do we have any questions? I guess there are no questions. Thank you. Thanks a lot. So I guess with this, we have come to the end of today's webinar on Vision Science and Optometry. So I hope everyone have gained some knowledge about the other researchers and also their topics. So we really thank you all for all of your patience, and every one of your presentation was really very neat and clear, and we really enjoyed. And I personally got to know many information on this, and thanks a lot for that. So I really hope to meet you all one day once this pandemic ends. So yeah. So thank you. Thanks a lot for your presence, and your participation means a lot to us. And we hope for continuous support for even our future events and everything. So if you're having any queries or questions, please do reach us out, or you can directly contact our program manager for any assistance. And regarding the certificates, you will receive an email with the conference, like the abstract proceedings book, and also the certificate through email, maybe within next 10 to 20 minutes. So thank you once again.
Thank you very much.
Thank you.
Thank you very, very much. Bye-bye.
Thank you. Bye-bye.